Let's look at photosynthesis in this section. And first point is, yes, there are bacteria that carry out photosynthesis. Not all bacteria carry out photosynthesis, but there are some. Okay, so let's go down through here and look at a few odds and ends. So the metabolic pathways that we've been looking at have been the breakdown of materials, breaking down carbohydrates, breaking down lipids, breaking down proteins in order to release the energy stored in those molecules and transfer that energy to ATP. What we're looking at with photosynthesis is a means of producing those organic molecules. So organisms are going to synthesize complex organic compounds from simple inorganic substances. And this is going to be photosynthesis. Okay, so you can notice right here, photosynthesis is a conversion of light energy from the sun into chemical energy. And then once we have that in chemical energy, that chemical substance containing the energy can be passed on from one organism to the next, or it can be used by the organism that made that. Okay, so let's take a look right here, and we have this term carbon fixation. Carbon fixation is conversion of gaseous carbon dioxide. So just imagine you've got these carbon dioxide molecules all just bouncing around in the atmosphere. Gaseous carbon dioxide into organic molecules. So now the carbons are locked into place, or that is, they are fixed. Fixed does not mean repaired in this case. It means fix as in to affix, to glue, to stick. So the carbon molecules are now glued into place or stuck into place. And notice down here, cyanobacteria, algae, and green plants all have essentially the same type of photosynthesis. Okay, let's pick up here. So plants, algae, cyanobacteria are all going to use water as their hydrogen donor. They're going to release oxygen. So right here. So notice what's going to happen in this process. The carbon, this carbon is going to be carbon right here. Okay. These oxygens are going to be right here and right there. Okay. Now then, this hydrogen, we've got 12 times 2, 24 hydrogens. These hydrogens are going to be the hydrogens that are on this sugar, and the oxygen right here is going to be the oxygen that is released. So 12 times 1 is 12, 6 times 2 is 12. Okay, now these hydrogens are also the hydrogens that make this new water. So the main thing is right here, water. So let's go right here, water, H2O. This is our hydrogen donor, and the oxygen is then released. Hydrogen right here that ends up in the sugar comes from the water. Now let's look down here. Purple sulfur, green sulfur bacteria, these are going to use H2S as the hydrogen donor. So hydrogen sulfide. Notice the similarity between H2S and H2O. Okay, but guess what's going to happen? It's going to produce sulfur granules. So here's carbon. It's going to be the carbon on the sugar. The oxygen from this carbon dioxide is going to be the oxygen on the sugar, as well as oxygen water. Now then, the hydrogens right here are going to come from the hydrogen sulfide. So hydrogen's in the sugar as well as hydrogens to make that new water. Now then, what's left over is that sulfur, and the sulfur is right here. Well, oxygen is a gas, and so oxygen as a gas just diffuses out of the cell. Sulfur is not going to be a gas, so the sulfur is going to wind up just accumulating in the cell as granules of sulfur. So, purple sulfur bacteria and green sulfur bacteria. And again, these are photosynthetic, just like these are photosynthetic. Okay, so let's go back over here. Let's go on down and take a look. So electrons are going to be taken from the hydrogen atoms, and we're going to incorporate that into the sugar. Now then, we've got this term right here, whoops, bring it back. There it is. We've got this term right here, photophosphorylation. Well, remember, phosphorylation is process attacking a phosphate group onto a molecule. For example, taking ADP, tack a phosphate on there, make ATP, that is phosphorylation. Okay, in aerobic respiration, as well as anaerobic respiration, we used oxidative phosphorylation, and the oxidation of those hydrogen carriers, NADH, FADH, the oxidation of those provided the energy to manufacture ATP, to tack that third phosphate onto ADP, making ATP. So oxidative phosphorylation. Now, in photosynthesis, we get photophosphorylation. Photo means light, and so photophosphorylation is where the energy from light, or let's say it differently, light is going to provide the energy to tack that phosphate group on to ADP and make ATP. Okay, so let's go on down here. Let's see. In this process of photosynthesis, we are going to have an electron transport chain similar to what we had in respiration. And we're going to have electrons that are passed along through these carriers. It's going to pump hydrogen ions across a membrane. Those hydrogen ions are going to come flowing through a protein complex called ATP synthase, which that's going to be processed chemiosmosis, and that is going to produce ATP. So we're going to have a process in photosynthesis that is very much like that last stage of aerobic and anaerobic respiration, an electron transport chain and chemiosmosis. Okay. Now then, we're going to have cyclic and non-cyclic photophosphorylation. So it's right here. Cyclic photophosphorylation. We're going to have light picked up by a pigment right here. Let's see. Light picked up by a pigment, chlorophyll. It's going to get the electrons going. Those excited electrons are high energy electrons. So basically, you might think of it like this. Energized, let's do this, energized electrons. These are going to be passed through an electron transport chain. As that occurs, this is going to pump hydrogen ions across a membrane. These hydrogen ions will come flowing through this ATP synthase right here, and that will convert ADP plus phosphate over here to ATP. Now, what happens here where it's cyclic is the same electrons are passed. So here's these electrons. These electrons are passed back to the chlorophyll and then recycled. Okay, so this process of cyclic electron transport, it does not So basically right here, it does not produce any oxygen, and it does not produce any sugar for long-term storage of the energy. What it does is it produces ATP, and remember, ATP lasts only for a few seconds before it breaks down. So ATP is very unstable, and it is used to provide energy to do work in the cell right now. It's not going to be stored for later. Okay, let's go up here and look at what we have. Now then, non-cyclic photophosphorylation. We have light being picked up by pigment, chlorophyll. The electrons from the pigments are highly energized. They're boosted up to higher levels. They're picked up by electron acceptors, passed along a pathway, and we're going to get right here, we're going to get here's our ATP synthase. So we're going to have our hydrogen ions pumped across the membrane. These hydrogen ions are going to come flowing through the ATP synthase. And when it does, it's going to convert ADP and phosphate into ATP. Now over here, notice they don't really show that one with the wavy lines like over here. So over here, we don't have the production of on the right-hand side, we don't have the production of ATP, but this electron is picked up by, so here's the electrons right here. That's going to be picked up by the NADP, and the NADP is also going to pick up this hydrogen, and it's going to form right here. So now what's going to happen is this hydrogen carrier is going to go to the next set of reactions and is going to use that hydrogen. So that hydrogen will be tacked here into the carbon dioxide to make right here C6H12O6, make sugar. And this ATP that's produced here or here is going to be sent over to this next set of reactions to provide the energy to drive that next set of reactions. Remember, all the work that's done in a cell, the energy for that work comes from ATP. When we're talking bacteria, we're talking animal, we're talking plant, fungus, whatever. All the energy for cellular work comes from ATP. Okay, let's go back over to the textbook. And let's see what we have. There's what we just looked at. And let's see. The second part of photosynthesis is called the light-independent reaction, also called the Calvin-Benson cycle, named after the individuals who worked this out. And this is where the carbon dioxide is fixed. That is, it's changed from a gas bouncing around out in the atmosphere to it's changed from a gas bouncing around out in the atmosphere to being locked into place in a sugar. Okay. Basically, carbon dioxide is going to come in. It's going to go through a number of chemical changes right there. We're going to use the energy in the form of ATP from the first set of reactions to restructure the molecules. We're going to drop off the hydrogens that were captured from the water in the first set of reactions. And then down here, we're going to make glyceraldehyde phosphate, which we saw in the middle of glycolysis. And most of that glyceraldehyde phosphate will wind up becoming glucose. Okay. Then we're going to have a few restructuring of the molecules here to get us back to our starting place so we can pick up more more carbon dioxide. Okay, let's go over here. Summary of energy production mechanisms. Okay, organisms are going to obtain energy from oxidation reactions. Okay. And so to be able to obtain the energy, cell has to have an electron or hydrogen donor, and that's going to serve as the initial source of energy. Now that's kind of a technical way of saying, well, in process of respiration, sugar is going to, what's going to be used from that sugar is going to be the hydrogens. What's going to be collected off the fats are going to be the hydrogens. So it's the hydrogens that are going to be used in this process of respiration, not the carbon chain. The carbon chain is going to wind up as carbon dioxide. Those hydrogens have energy-rich electrons in there, and those electrons are going to drive the process that's going to allow ATP to be made. Now then, you notice down here, the other one, they talk about photosynthesis. Well, the photosynthetic pigments could be our electron donors. So the photosynthetic pigments absorb light energy. It gets their electrons going faster, 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 faster. And then those electrons can be passed on to the production of sugar. Okay, then we're going to have hydrogen or electron carriers, and that's going to be our NAD, NADP, and FAD. Okay, requirements for ATP production. So notice what we're going to have. We're going to have either photosynthetic pigments in conjunction with light, or we're going to have glucose or some other source of hydrogen. Okay, some other source of hydrogen or electrons. And so from that flow of electrons, we can produce ATP. And then those electrons can be picked up by electron carriers or hydrogen carriers, which are going to be the NADP in the case of photosynthesis, NAD, FAD in the case of respiration. And then we're going to have some kind of electron acceptor at the end. Okay, so these electron acceptors can be, and in case of respiration, all of those down at the bottom are talking about forms of respiration. The electron carrier could be oxygen in the case of aerobic respiration. It could be some compound other than oxygen, such as nitrate or sulfate, as in anaerobic respiration. Remember, anaerobic respiration, you still have an electron transport chain, and you still have chemiosmosis, and you still have an electron acceptor at the end of the electron transport chain. Also, with anaerobic respiration, you don't have oxygen involved, but these other electron acceptors like nitrate and sulfate are just not as good at picking up electrons as is oxygen. So the in bacteria, you can, from one glucose molecule using oxygen in respiration, aerobic respiration, you can get 38 ATPs. But in bacteria that carry out anaerobic respiration, you will never get 38 ATPs. It will always be something less because your electronic acceptors at the end of the electron transport chain, nitrate and sulfate, are just not as efficient as oxygen. Okay, the other possibility is the electrons, and thus the hydrogen atoms, are picked up by some kind of organic compound, which leads to the production of ethanol, lactic acid, or a number of other possible compounds that we saw earlier. Let's see if there's anything that we need to look at here. I don't see anything that jumps out at me. So let's see if we can go on over here. Metabolic diversity among organisms, microorganisms. Oh, that's a whole topic in itself, so let's just stop this one right here.